Welcome back on my channel. In this video, I will introduce about principle and processes. Right. Tools of the recombinant DNA technology. इसके बारे में हम लोग पढ़ने वाले हैं. Right. Now we know from the foregoing discussion that genetic engineering or recombinant DNA technology can be accomplished only if we have the key tools: restriction enzyme, polymers enzyme, ligases, vectors, and host organism. Okay, let us try to understand some of these in details. Now we will see one by one. The first one is restriction enzyme. In the year 1963, the two enzymes responsible for the restrictions, the growth of the bacteriophage in Escherichia coli were isolated. One of these added methyl group of DNA, while the other cut DNA. The latter was called restrictions endocloses. Right. Next, the first restriction endocloses hind second, whose functioning dependent on a specific DNA. Okay, nucleotide sequence was isolated and characterized 5 years later. It was found that hind second always got DNA. Right. Molecules at a particular point by the recognizing a specific sequence of six base pairs. This specific base sequence is known as the recognition sequence for Hind second. Besides Hind second, today we know that more than 900 restrictions enzymes that have been isolated from over 230 strains. Right? A bacteria, each of which recognize different recognition sequences. Right. The convention of the naming these enzyme in the first letter of the name comes from the genus, and the second two letters comes from the species of the prokaryotic cell from which were isolated. Okay, E. coli comes from Echoria coli RY 13. In E. coli, the letter R is derived from the